One of the important things in science and scientific observations is making predictions and then seeing if those turn out to be true. And for evolution, it turns out so many of its predictions have turned out to be false. And yet, evolution still persists as a scientific theory in spite of many, many false predictions. Now, I'm here with Dr. Cornelius Hunter, and he has identified and written about 23 false predictions that evolution has made. Today, we're going to be talking about false prediction number 7, the molecular clock. Welcome, Dr. Hunter. Thanks for being here.
Oh, thank you, Rebekah. I appreciate you inviting me. I'd love to be here. So
Thanks.
Let's bring up those slides. And you mentioned 23 false predictions. I kind of ran out of gas at that point. It's not that there's only 23, but there are a couple of dozen key at least a couple of dozen key fundamental predictions of evolution. And so today we are on number 7, the molecular clock. And you cannot talk about the molecular clock without talking about Linus Pauling, a great American scientist, a great American chemist. 20th century, his dates put him squarely in the 20th century, 1901 to 1994. And Pauling was a chemist. He correctly predicted the globular protein secondary structure. There are 2 main secondary structures that we find dominating globular proteins. There's an alpha helix and beta strands, beta sheets. He predicted all that, so that was pretty good. He also did some key work in chemical bonding. And that was really his fundamental work in chemistry, the modeling of chemical bonds. And that earned him a Nobel Prize. So he's a Nobel Prize winner. He was chair of the chemistry department at Caltech. So really on top of the world. He also made another prediction about organic chemistry, and that was the DNA. So we all know about the DNA double helix. Well, before we knew about that, and that was a big race to figure out what exactly is DNA, what is the structure? And you know that Watson and Crick got the double helix. Well, around that time, Pauling was thinking about that, and he came up with a triple helix. So, and also he had the side chains pointing outward. Now that made all the sense in the world because you have to read the side chains of the nucleotides in order to read the DNA. So yeah, why not have them pointed outwards? That makes sense. Well, it was wrong. It's a double helix, not a triple helix, and they're pointed inward, which it doesn't make a lot of sense, but that's the way it works. That's the way God did it. In order to read those, you have to pull the double helix apart, and there's proteins that do all that. Crazy. So it's an incredible system. So we got that one wrong. Got a couple right there. He also did a couple other things. He was a peacenik before his time in the early 60s. When it really wasn't, you really weren't into the peace movement yet. Yeah, he was. And so that was a little bit controversial. And they eventually booted him out of Caltech. I mean, not literally, but they made it difficult for him. But he did get the Nobel Prize. So he's one of 5 people in history so far to have 2 Nobel Prizes. So good job, Linus Pauling. But there was another thing that kind of tarnished his career, which was he really got into vitamin C in the latter part of his career, and he had a many years studying this and at an institute. In his legacy, he has a research institute in Northern California, so that's good. But the vitamin C thing turned out to be he was a little overconfident, very overconfident actually, and it didn't work out the way he thought it would. Now today, we actually kind of tilting back towards his position that we do think vitamin C is pretty good, but probably not as good as he thought it was. So anyway, he did die at the age of 93, so vitamin C probably couldn't be all that great, right? So a very interesting character. And when you talk to somebody about Linus Pauling today, they're only going to remember the vitamin C part. Right? That's the unfortunate thing about his legacy. You think of Newton, and that's the universal law of gravity, and Darwin, that's evolution, Einstein, that's relativity, Pauling, oh, vitamin C. There's so much more to Pauling than vitamin C. And so that's unfortunate. And so good historians or people in know, they understand these other things, the alpha helices and the beta strands and the chemical bonding and the 2 Nobel Prizes and, the peace activities, all these good stuff. There's one other thing that most people do not know about Linus Pauling, and that's what we're going to talk about today, which is the molecular clock. Linus Pauling is the guy who came up with, in the 1960s, the concept of the molecular clock in evolutionary theory. So he was an evolutionist, and he came up with this idea of a molecular clock with some papers that he co-wrote with Emil Zuckerkandl. So what is this molecular clock? So here's an evolutionary tree. The top part of that evolutionary tree are the extant species. So there's a couple of extant species in those yellow circles. They have a common ancestor, the blue dot. How long has it been since the blue dot? How long do you have to go back in time? That's one of the things that evolutionists spend their time figuring out. How long are these branches in time, right? So one way to do that Pauling came up with, of course, there's the fossils. You can look at the fossil record and do some dating in the strata and figure that out. But that's got its own vagaries, as everyone knows. But what Pauling noticed was if you look at proteins that are in these extant species, you can nail down the same protein and think about, okay, that protein evolved from a common ancestor in the blue dot. But the proteins in the extant species are not identical. They have some amino acids that are different. If you look at the gene, there are nucleotides that are different. Well, what's going on there? Well, they've been mutating. And they've been mutating at some kind of roughly constant rate. It's stochastic, but it's kind of, mutations happen, in the aggregate at an average rate. And so you can actually tally up how long it's been according to how many mutations they have. Now, let's zoom in here. So you want to know how long this branch is. Well, you compare the 2 yellow dots, the protein that's in the 2 different species, and look at how many differences, how many nucleotides are different. Under evolutionary theory, those occurred via mutation. So you count up the number of mutations in those genes and then say, what's the mutation rate? You can figure out how long it is. Molecular clock. That's the idea. Common ancestor. So for example, a cow and a horse, consider cytochrome C, a heavily studied protein. Look at the gene, count up how many differences there are. Now, here is a chart that evolutionists like, and this is the cytochrome C chart. So the dots, the red dots, show the number of changes in the cytochrome C gene between 2 species pairs. So there's different species pairs on that plot. So each red dot is a different species pair. The first one is a horse and the donkey on the left-hand side there. Not very many changes because the horse and the donkey are so similar. Their common ancestor is relatively recent. Not very much time, not very many mutations. And then you go out to more and more differences, the horse and the cow. Well, that's a big difference. Should be bigger changes. Should be more mutations. That's exactly what we see. Hey, this looks good. So they line up. Look at all the dots line up on this line like really well. This looks really promising. So there's the horse and the donkey, and you can look at the time. It's the number of mutations there is like 10. And if you assume a certain rate, that's going to map into a few million years. And then you can look at the horse and the cow. Oh, okay, that's a lot more mutations. Now you're up to 50 mutations, 50 substitutions, 50 differences, and that's going to map into a longer time, like 70 million years. Then there's the horse and the cow. More substitutions, more differences, a longer time. The molecular clock. You're basically using these mutations, using the differences in the genes as a clock, as a way of measuring time. And that concept really caught on. Evolutionists over time, and this would be an incredibly high impact paper if you measure the number of times this has been referenced in the literature, the number of times the molecular clock concept has been used by evolutionists. It's incredible. It's a super high impact paper and high impact concept. I'll just read you some quotes from some evolutionists. The discovery of the molecular clock stands out as the most significant result of research in molecular evolution. One of the simplest and most powerful concepts in the field of evolution. Roger Lewin. Zucker, Candle, and Pauling showed that many, perhaps most molecules have a rather constant rate of change over time. Such molecules can serve as a molecular clock. Ernst Mayr was one of the leading evolutionists of the 20th century, a Harvard professor. So very high quotes here. Here's another paper referring to it as a discovery. So if it's a discovery, it's something real. There really is this thing of a molecular clock. There's something was discovered. So this isn't just like some fake model or something. So the prediction is there is a molecular clock for past speciation events. It may not be exactly perfect. There's going to be some noise. This is not predicting, perfect 3 decimal estimation of time, but it is pretty good. And it is a clock that can be used. Well, it has been falsified, and I'm not talking about just a little bit of precision loss. This is major fundamental falsifications over and over and over and over again. And I'll quickly run through the different ways. Let me count the ways that the molecular clock has been falsified. So first of all, different protein genes must evolve at different rates. Now, what does that mean? So I showed you that cytochrome C chart a minute ago. Well, on this chart, that's the orange line, okay, cytochrome C. Now you'll notice there's a couple other lines. There's a hemoglobin, and there's a fibrinopeptide. Well, what's going on there? Well, those are different proteins, and there's different genes coding for those proteins. So each one has its own, gene. Well, they all show different rates of change over time. So the dots, if we were to show the dots, yes, they would line up in more or less a line, but they're different lines. And so there's radically different rates of change. If the molecular clock is true, there isn't a universal rate of time, rate of mutation. They're radically different between different types of proteins. So that's interesting. What's going on there? Now, of course, evolutionists do have their explanations for this. They tick at different rates because these different proteins can tolerate mutations differently. Some are more sensitive to mutations. They're not going to allow as many mutations. So they have different rates. Okay, so in this quote here, it says, some proteins are what? Highly constrained by natural selection. That's the outcome of this. Okay, the prediction was false, but it's okay because we have these different rates for different proteins. So that's the explanation. The outcome is, different rates. Because some are highly constrained, some are relatively constrained, some are highly constrained by natural selection. Okay, where did you figure that out from? What is the evidence? What is the experiment? Well, how can we know that? Oh, there is no evidence for that. That is an inference, assuming evolution is true. So the epicycle that we're attaching here, the explanation that we're using to explain this is based on the assumption that evolution is true. So that would be circular. So, okay, that's not so good. This is assumed in order to save the theory. Okay, now here's a similar plot. The only difference on this graph, this is from Thomas Jukes, there's another protein on here. So there's 4, not 3. You got the fibrinopeptide, you got the globin, you got the cytochrome C. There's a histone 4 there, real close to the x-axis, the horizontal axis. That one is incredibly constrained. We talked about that in another false prediction. Very highly conserved protein. So that's even more awkward. It's like, oh, wow. We also looked in that other false prediction that actually is not highly constrained. So Michael Behe had done experiments for one example. There's many other labs that have experimented with this that showed that histone 4 actually isn't that sensitive to mutations, and it does have some wiggle room, and you can make changes. You can't make a ton of changes, but most proteins do not sustain very many changes. But what the molecular clock is claiming and insists and must have is that very, very, very few changes, even single changes to histone 4, can be sustained. Most mutations are fatal. You cannot sustain most mutations, single mutations in histone 4. Well, that turned out to be false. So that didn't work out very well. So we got that problem, histone 4 right there. And there's my video, false prediction number 6 on that. And we also did a video on that. Okay, so that's one problem, but there's another problem. So the same protein, but different lineages must evolve at different rates. So we just looked at different proteins, but what about the same protein, but in different parts of the evolutionary tree? So different lineages, cytochrome C or hemoglobin, globin, they have different rates. They must, if the molecular clock is true, if evolution is true, they must have different rates. So in other words, when you look under the hood and you start looking at the details, you realize, oh, in this lineage over here on the evolutionary tree and this lineage over here, they're behaving differently. So here's a paper on that. Different types of protein diverge at different parts of the different rates on the evolutionary tree. So not a good thing. And let me blow up my screen here a little bit. That's better. Yeah, different types of proteins diverge at vastly different rates. Moreover, because that's what I just said earlier, different proteins at vastly different rates. Moreover, the same type of protein has been observed to evolve with different rates in different phylogenetic lineages. Not good. Now, you'll notice that phrase there, has been observed. Has it really been observed? The same type of protein has been observed to evolve with different rates. Oh, wait a minute. Where did we observe that? This is another inference from evolutionary theory. So if evolution is true, then we believe that's what happened, right? So this has not been observed. This is a model-driven inference. Must be true if evolution is true, not observed. So this is what you get. This is why evolution harms science. Evolution is not good for science because it is always doing this. You're bringing in, you're looking at science through the evolutionary lens and then documenting it and describing it as just an observation. Well, no, it isn't an observation. So this is a phony science. Strong and lineage specific regional effects were seen on the molecular clock. So it's also geographic. It's, as you might expect, different parts of the evolutionary tree, different geographic regions. So it's lineage specific, it's geographic specific, and again, were seen. So these different rates were seen. No, they weren't seen. They're inferred. This is an evolutionary inference. You have to keep that straight in science. It's crucial in scientific theory. You have to keep this straight. And it doesn't. Unfortunately, this is not. We don't see this in the evolution literature. So the failures we've just looked at: the protein type. The molecular clock is not universal with respect to the protein type, the taxonomic lineage, and the geographic region. Okay, what else? Anything else? Protein genes must evolve at different rates over time. So there's also a temporal change that must be happening. Estimating, here's a...
I'm starting to get the feeling there's no clock here, right?
Well, we're gonna, I think you're anticipating. You're anticipating what's coming here. But check out the title of this paper. Estimating the rate of evolution of the rate of molecular evolution. A clever little title. In other words, the rate of molecular evolution is changing. So they're estimating the rate of evolution of the rate of molecular evolution. Very clever. But right there, you've got it right there in the title. Oh, so this isn't working. It's changing over time. So now we have time added to our list. What else? It's contradicted by the fossil evidence. So that would be a real good way to check this concept of a molecular clock, would be to compare the molecular clock with the fossils. Not that the fossils are always right, but we should be able to at least have some congruence, and we don't have that. In fact, we have big differences. Large discrepancies have been found in the dates of evolutionary events obtained using the molecular clock. Twofold differences have been reported between the dates estimated from molecular data and those from the fossil. What? Twofold? Yeah, this is not like, third decimal point differences. Now, here is another paper that actually, Rebekah, you provided this one to me. And another great example, these are plants. Now, this comes from the nightshade family. The nightshade family has all kinds of interesting secrets and tidbits that contradict evolutionary theory. in this case is the molecular clock does not match up with the fossils, and by a huge amount. This is way more than twofold, by the way. This is a huge discrepancy. The fossils are considerably older, like on the order of 50 million years is the estimate when you look at the fossils, than the corresponding molecular divergence dates, which are on the order of like 10 million years. So that's a great one, Rebekah. Thank you very much for another example to illustrate these problems.
Hey, all that came from me just looking up an article about how did tomatoes evolve. And I just happened to run into this, and that's why I love looking up how things evolved, because every time it's like whatever paper you get about how something evolved, it just so beautifully demonstrates the problems with evolution.
There are always nuggets.
Yep.
There are always nuggets. Yeah, you find out, it's just, oh, well, what's going on here? Another nugget with the nightshades is the embryonic pathways, the development pathways. They don't fit. That's a whole nother story. Here's another example. Now, this is from primates. Yeah, so another example where the primate fossils and the molecular clock didn't match up.
Okay, so in this case, they're doing it they're not doing it with the proteins, they're doing it with the overall changes between organisms. Like, don't they count it up that way too? Like they do their molecular clock on the amount of total genetic differences, or am I missing something there?
Yeah, no, that may be right. I can't remember what the details on this one are. If this was an aggregate over large parts of the genome, or if this was based on specific proteins, specific genes. So yeah, it could be. There are many ways to apply the concept of the molecular clock. 31 species. Yeah, I have to go back and look at that one to get the details. But the global molecular, I see global molecular clock, so that you may be right about that. The global molecular clock was clearly violated. Yeah. Next example. So in addition to the fossil problem, there are different molecular methods. There's, as you might imagine, there's different ways of modeling the evolutionary process at the nucleotide level and so forth. And they produce different results that don't make sense, that are not inconsistent on the molecular clock. Different molecular methods can give dates that differ 20-fold. So that's an enormous internal contradiction within the theory of evolution. Just the different ways that you can model these things really are inconsistent. The research implies that many previous studies, even some of those using recently developed methods, might have placed too much confidence in their date estimates, and their conclusions might need to be revised. Yeah, so that's a good point indeed. So the molecular clock is internally inconsistent. What else? Well, we can observe mutation rates, and now we're inferring mutation rates from the molecular clock because we're lining it up to the fossil record, and we're saying, okay, what must the mutation rate have been in the long run, and they don't match up. So there's a big discrepancy between observed mutation rates that we can observe in the lab or in the field and what the molecular clock implies. So here's a paper that talked about that. Studies of molecular evolutionary rates have yielded a wide range of rate estimates for various genes and text. Okay, we just talked about that, how there's these different rates. They're not always consistent. Recent studies based on population level and pedigree data have produced remarkably high estimates of mutation rates. So these are more things that we can trust. These are not based on evolutionary theory, which strongly contrast with substitution rates inferred in phylogenetic studies. Now here you see they use the word inferred. That's science. You know you're reading a good science paper, or at least you have the hope here, because they're pointing this out. What is inferred and what is observed? And so that's nice the way they did that in this paper. Our results show, then they have to, because they're pointing out this discrepancy. Our results show that it is invalid to extrapolate molecular rates of change across different evolutionary timescales. So again, the molecular clock is failing. So we got molecular clock contradicted by observed mutation rates. What else? Well, different proteins contradict each other in more bizarre ways. And this is a good paper. There's two proteins in their genes that were particularly contradictory: glycerol 3-phosphate dehydrogenase and superoxide dismutase. And so this paper was going to look at those two genes and really try and figure this out. And then to try and help with their test and their comparisons, they brought in another protein, xanthine dehydrogenase. So we're looking at 3, but that third one was kind of the outlier that they're going to use to try and help. Well, the whole thing kind of blew up. They not only confirmed these differences, but the xanthine dehydrogenase didn't help. It just brought in more differences. So it's really kind of hilarious. It has been observed that the variance of the rate of evolution is generally larger than expected according to the neutrality theory, which has raised the question of how reliable the molecular clock is, or indeed whether there is a molecular clock at all. So we're getting there to your earlier comment, Rebekah. We have carried out an extensive investigation. So this is not just a little study here of these 3 proteins. We have observed that the 3 proteins evolve erratically through time and across lineages. So it's not working. Skipping down here. Now, there's a lot more in this paper, and I just didn't want to be reading. So I'm skipping down, but you can look at this paper. But I'll skip down with this kind of conclusion type result. We are left with hypotheses that attribute rate variation to the vagaries of natural selection. So that's really when evolutionists invoke natural selection to solve these molecular clock problems, it's kind of like they're giving up. Because when you invoke natural selection, then anything goes. You can claim anything. Oh, well, there was natural selection. It had to do that. And so you're really kind of losing the concept. Whether as a consequence of functional shifts that change rates of neutral mutation through time differently for different genes in different organisms, or in response to phenotypic evolution and environmental heterogeneities, these 2 hypotheses amount to a denial of there being a molecular clock. So Francisco Ayala. He's it's his lab here at UC Irvine that's doing this work back in the 2000s. And yeah, he really kind of came down on that there really is no molecular clock. So another failure, molecular clock is contradicted by observed. Oh, I didn't have it on the slide, but there's another one, which is, they're just different genes are just contradictory. I mean, if I went into the details of that paper, you see some protein, one of the proteins shows a different rate in one part of the protein than another part of that same protein. But then in that same lineage, the other protein shows like the inverse trends. So just really all over the map. It's erratic, as they said. I didn't want to get into all those details, but it's very significant. So overall, we have a falsification of the molecular clock in many, many different ways. It's just absolutely failed. Oh, now this paper kind of steps back and looks at the big picture and just says, yeah, numerous follow-up studies, including ours, have falsified the concept of a universal molecular clock. Work on fossil sequences also invalidated the molecular clock, so the fossils did not agree. So that's a representative look at the literature on what happened with the molecular clock prediction. It failed and failed over and over and over again in many ways. So what's the outcome? So there's several bad things that come out of this. And I've talked about this before on these false predictions. It's important when you have a prediction and it turns out to be false, that can be a good thing for a theory, depending on what is the outcome. You can learn more. You realize, oh, we had a bad measurement. We had a false understanding of the problem, the scenario that we had, the data that we fed to the model. Something was wrong in the way we were feeding the model. Or maybe the model itself needed to be updated. We're not tossing the whole theory out, but we're adjusting it. Maybe we're going to make it more simple. We now realize, oh, it's even better than we thought. We can reduce the complexity of our model. This is a great ending. This is a great outcome. Those are the things you want to look for: reduction in complexity, increase in parsimony, or some error in your data, error in your understanding, something like that. that didn't happen. It doesn't generally happen with these false predictions in evolution. What you get is bad things coming out. So one of the bad things here is a loss of parsimony. In other words, the evolutionary models became more and more and more complicated. And I'll just share a couple of examples. Most workers now agree that a global molecular clock does not exist, and that rates can change between lineages and within a lineage over time. In other words, like that just covers the waterfront, right? New methods of estimating branch lengths in time do not assume a strict clock, but the number of models for molecular evolution is then what? Effectively infinite. So once you drop the idea of this, the restriction that there is this universal molecular clock, but instead, well, it can be this, it can be that. Where are you going to stop? Where do you draw the line? Where is your red line? There is no line. So it's effectively infinite. Neer and Sanderson noted that, quote, with respect to rate heterogeneity, once the model of molecular evolution departs from a simple one-rate molecular clock, the number of models is what? Effectively infinite. So complete loss of parsimony here. Another problem, another negative outcome is the loss of the underlying fundamental evolution model. So this idea that of the molecular clock and what Pauling came up with is really not totally unique or new with Pauling. And the basic idea goes back to Darwin, that the rate of change accumulates over time. So the more change you observe between two different species reflects on how long they've been evolving. So it's a kind of a basic concept in evolutionary theory. And that's what this paper is talking about here. Authors such as Hutton, 1872, have concluded instead that differentiation of form, even in closely allied species, is evidently a very fallacious guide in judging the lapse of time. So they were finding out about this, way back then. There's this fundamental problem. Croizet in 1964 also criticized the universal clock approach and made the opposite assumption, namely that different groups in the same region, like New Zealand or South America, can and do usually show tremendously different rates or at least degrees of differentiation during a single phase of evolution. So not only is it like not quite working, here we're saying, oh no, it's the opposite. This is like a total train wreck here. Another bad outcome was the loss of the fundamental premise of clock-like rates. So this is a great paper here, and I can't read through all the juicy passages, so I'm just going to summarize it, but it's a great paper to look at. But I do just look at the title, The Dawn of Relaxed Phylogenetics. Nice title, 2023. Now, what does that mean, though? Relaxed phylogenetics. So what is phylogenetics? Phylogenetics is the study of the evolutionary tree. So developing numerical methods, developing tools, developing concepts, or coming up with results of what is the evolutionary tree? How can we improve it? What's the latest version of the evolutionary tree? Usually evolutionists work on some limited part of the tree, like this family or so forth. So phylogenetics is a general term for the study in evolutionary theory. Well, relaxed, what does that mean? Relaxed phylogenetics. Well, because nothing is working, we're going to relax those rules. And instead, we're going to allow for all kinds of knobs and levers that we can twist and turn. So the model becomes highly parametric. So you can adjust this thing like crazy. So I'm just going to summarize this paper in these 6 bullets because it would be too much to read. But first of all, they start out saying, hey, Pauling's molecular clock 8, that was a great idea. Nice job. But the molecular clock has failed big time. So it is a substantial problem in evolutionary theory to try and figure out what is the evolutionary tree. So we have a new thing that we call statistical phylogenetics or statistical phylogenetic methods, and they fall into this broader category of relaxed phylogenetics. And in this statistical phylogenetic methods, in these methods, each branch can have its own rate. So if you look at this huge evolutionary tree and all these species, and each branch can have its own rate. That is like the ultimate in tautology and in the loss of parsimony. In the entire evolutionary tree, you just got a gazillion different knobs. You can play with each one. And of course, they're going to go further than that and allow for different proteins. They already have done that within the branch, different genes, protein coding genes, they also have their own rate. So they're all over the map here. So it's an incredible loss of parsimony. And then as a conclusion, they say, yeah, in all this work, we've kind of confirmed that most genes exhibit non-clock-like rates. So yeah, we found that out. Yeah. Okay, another negative outcome was the multiplicity of explanatory mechanisms, which just like all these different mechanisms to use to explain things. Determining the relationships among and divergence times for the major eukaryotic lineages remains one of the most important and controversial outstanding problems in evolutionary biology. Okay, so we got this problem, we're working on this problem. Yet, relationships among these groups remain poorly understood because of saturation of sequence changes on the billion-year timescale, possible rapid radiations of major lineages, phylogenetic artifacts, and endosymbiotic or lateral gene transfer among eukaryotes. In other words, this is not a complete list, but you've got right off the bat half a dozen different explanatory mechanisms that can be drawn on to pretty much explain anything. Error in such analyses comes from a myriad of sources, including calibration fossil dates, the assumed phylogenetic tree, the nucleotide or amino acid substitution model, substitution number, the branch length estimates, the model of how rates of evolution change over the tree, error inherent in the time estimates for a given model, and how multiple gene data are treated. Again, a mouthful. The point is there's all these different possible causes of error and estimation problems. And, so you've just got a non-parsimonious theory. Okay, let's just by reanalyzing data sets from recent published molecular clock studies, we show that when errors from these various sources are properly accounted for, so they're saying in the past, we've got all these error sources, they haven't always been accounted for. Now we're looking at these, we're accounting for all these different possible errors for us. What we're finding is that confidence intervals on inferred dates can be very large. Now, when something is very large, when confidence is very large, that sounds like a good thing. Oh, wonderful. Evolutionary theory is getting more tight and it's looking really good. No, this is the opposite. Confidence interval is basically, it's a synonym for the uncertainty. So the uncertainty is getting very large. So this is not a good thing. This is a bad thing. Okay, let's look at another illustration of the multiplicity of explanatory mechanisms. I won't show too many more of these, but it's just kind of interesting. Molecular dating has now been adopted in many areas of biology. This broad uptake has been due partly to the development of Bayesian methods that allow complex aspects of molecular evolution, such as variation in rates of change across lineages, to be taken into account. Okay, so we've got these Bayesian methods now we're going to bring to bear. These methods rely on assumptions, quote, which vary in their degree of biological realism and empirical support. So that's what the highlighted quote there is. These methods rely on assumptions, what? Which vary in their degree of biological realism and empirical support. Now, when an evolutionist says, well, this varies in its degree of biological realism, that's a euphemism for saying it's not realistic. This is not biologically realistic. This is a made-up modeling. It's a numerical method and a modeling technique, has nothing to do with biology. So how are we going to write that up? Well, it varies in its degree of biological realism. This is not a scientifically accurate this is not science. Philosophers refer to realism and anti-realism. This does not fall under realism. It's a model that it has no correspondence with biological science. And they also vary in their degree of what? Empirical support. Oh, okay. So there's no empirical support for this. This is a model that you're using, which has no experimental evidence supporting it. So not very
So
good.
are you saying, like, when we're seeing people do all this math, like they're like, okay, according to this math and this math, this is the rate at which we should see like mutations happening, and this amount will be neutral mutations, and this amount will be this, and then this is the rate that we'll see change in the organism, that this is that what this is saying there's no empirical support for?
You're going down the right path, but you got to go a little bit further. They're attempting to fix the problem and to reckon with all these problems with these, the molecular clock, and so they're coming up with this new method called these new set of methods that fall under the category of Bayesian. So they're Bayesian methods, statistical methods. They're Bayesian, kind of like the earlier paper that we looked at. And so it's like, okay, good. You got some new methods to help solve the problems that we've been reckoning with for decades. Okay, great, wonderful. There is one little problem. These new methods we're bringing to bear, they're not really realistic. They don't really have they don't have a correspond to biology. They're not biologically realistic. And also there's no experiments or observations that we have to really, support these. So they're mathematical techniques to solve the problem, but they're not realistic. It's like, let's say you had the rate of change that just kind of jerked around in time. And so, well, we fit the data. I'm making that up, but I'm just to illustrate if it did something that was just not biologically realistic or observed. Now, this observed mutation rates contradict a molecular clock requires rates. So I may have mentioned this earlier, but the observed mutation rate that you can get from the laboratory is very different. I think we talked about this earlier, but it's very different from what you need to make the molecular clock work. So if you're going to calibrate the molecular clock to the fossil record, for example, it's going to imply certain mutation rates. Those are very different than what we actually observe in the lab. So this paper talks about that. I think I mentioned this. I'm going to skip over this one. We talked about it. So a summary of the outcomes that were not good that we just looked at: loss of parsimony, loss of the underlying fundamental evolution model, loss of the fundamental premise of clock-like rates, multiplicity of explanatory mechanisms, observed mutation rates contradict the molecular clock, required dates that are implied by the molecular clock are not consistent with what we observe. I mean, this is just all over the map, right? From every angle, this is just not working. And it's really a good indicator, a good a strong sign that the theory isn't working very well, right? So when you have a prediction, a failure, let's say, well, maybe that prediction failed and there's a way to tweak it or something. Maybe it's not the most parsimonious, but at least we're still hobbling along. This is just such a train wreck from all angles that it just shows this isn't just the molecular clock. I mean, these are fundamental concepts to evolutionary theory that are falling down one after the other. So it's a pretty significant failure of a prediction. And so I have my ending slide, as I usually use: religion drives science when it matters, because the reason why the theory sustains all of this, you might wonder, well, why are there evolutionists? What's going on here? Why do we still have this? And it's because it wasn't about the science. The science was doing the work that was driven to do by religious and theological doctrines. So theology is behind this. So you have to have the science tell the story, even if it's not working. You're going to use those scientific terms and concepts to fill in the blanks. And if they're not working, well, that's just the way it goes. We'll figure this out someday. So I have so many memories of lectures I've been in life science classes where the professor is explaining, this just incredibly ridiculous concept. This thing is, no way this could have actually evolved. but they're just explaining this as though it evolved and kind of with a grin on their face. Like, oh, that's the way evolution works. Isn't that amazing? Look at this. And no one had ever kind of asked the question, or the professor never asked the question, gee, I wonder if maybe this isn't the right theory. That just isn't brought up. So it's very interesting.
Yes. Well, now I want to give a little opportunity for pushback here. And so I think, the first thing that comes to my mind is, maybe somebody saying, well, this, and there are comments that I'll put up in a minute, but so somebody might say, well, even if the molecular clock doesn't work, that's fine. We can just abandon that. But, the theory's still intact. It's just rates are too changing. So we just can't, figure that out. Okay. So no clock, no big deal.
Yeah. So at the very least, what these are intended to do is to get everyone on the same page. And we can all agree, okay, the molecular clock prediction failed. It was a prediction. It did fail. Here are the outcomes. Without making it, we don't have to go further than that. We don't have to say the theory therefore is false. That certainly one false prediction does not falsify the theory. That would be naive falsificationism, but it does have implications. And the bad outcomes, as opposed to having good outcomes, is not a good thing for the theory. So it is an important failure. It's not a failure that was gracefully sustained. So can we just keep on going? Let's just keep on going here. So what? The molecular clock is problematic. Yeah, there's just all these different rates of change, and they're caused by all these different factors because the genes are all different and the species are all different and the eras are different. The geological strata are different. The different epochs of time are different. The cosmological rays were different, and everything is all different. So there's just differences happening all over the place. So yeah, that's what you're going to have to do to the theory. And so we at least need to understand and agree that that's what is being done with the theory. It is non-parsimonious. You're adding a lot of explanatory factors. And if you look at those papers, you can see the concessions that are made and the degree of explanatory factors that are brought to bear that are not biologically realistic and not empirically supported and so forth. So there's a lot of concessions that we need to at least acknowledge and say, yeah, this is a challenge for the theory.
Now, are most scientists involved with this admitting the molecular clock is either, have they admitted there is no clock? Are they admitting that it has problems? Or are they largely in denial about the problem?
I think what in my reading of the literature, most evolutionists in their papers use the evolutionary clock with an understanding that it's risky, and they will limit it to a particular small portion of the evolutionary tree, particular lineage, particular set of species, particular time frame, particular gene. And so if it kind of is helping them and it's helping them to tell their story and it's working, then they'll use it. If it's not working, then they won't use it. And they'll kind of use it guardedly. I don't think any of these scientists, evolutionists in print are naive about the problems. It's well known in the literature, so that no one is just going to come out and claim great things about it. But they do use it as a data source, kind of like the fossils, because we also know that the fossils have their own vagaries with the dating and so forth. And so with that, if you're an evolutionist, you're in a world of a lot of uncertainties with your measurements. But then on top of that, a whole bunch of explanatory factors that you draw from to make this thing make sense. Because the fact is, the dating of the fossils and the sequences of the genomics, the genes, the genetics, they're not that bad. I mean, you do have some pretty decent data to work with, but you're force fitting it into a model where it's not working, and so you're going to twist it and everything. So that's where the creativity comes in, and that's where all the explanatory factors come in. So I do see it being used, but being used cautiously.
Okay, that's interesting. And what I noticed too is that it can shift very the dates for these things can shift very dramatically. Like I saw this new discovery of tetrapod footprints that kind of pushed back the evolutionary timeline by like 35, 40 million years. And that just happened like last year. So, you know, these timelines and some of their molecular clock stuff is partially based on the fossils. So they, use the fossils and the genetics and they mix it all up and they try to figure it out. But then they can, a new fossil discovery can just change it by 35 or 40 million years. So, and this isn't the only thing I've seen like this. This is just the one example I seized upon. But I mean, you regularly see these articles coming out, oh, this timeline shifts, that timeline shifts. And so it makes you wonder, how scientific are those observations, those determinations in the first place, that they can shift so wildly.
And also, how well accounted for are they going forward? Is this going to be, safely contained within the repository of information, the knowledge bank of evolution? What you'd love to have is a history and an accounting of every paper, every study that was done where they came out and said, oh, well, we have to change this. We have to add this new lateral gene transfer, or some new mechanism, even a whole new mechanism that no one ever thought of. Oh, well, there's, and so this, I agree with you, Rebekah, that when you read the literature, you absolutely commonly run into this over and over again with papers. It's like, oh, well, here's this new thing we have to have. Like, is this deposited in some repository of the evolutionary theory? And so this is going to continue, because I like to compare it to the tax code. It is so complex. There's so many papers that make these statements that, well, we have to have this now. And you wonder, is this fully accounted for? Obviously, the more well-known, the more popular, the more famous papers and things are accounted for in later papers, and they are debated amongst evolutionists. But you wonder how many aren't.
Yeah. And would you say that, like, the rates that they're proposing, do you know in general, like the molecular clocks that they're proposing in general, are they proposing, it being faster or slower than what it turns out to be with our empirical observations?
The rates are too high. The empirical measurements that we can make in the lab are higher than the inferred rates from the molecular clock. And yeah, so there's something that it would have to be radically slow these things down.
Okay, that's interesting.
Over longer time periods. And I'm sure evolutionists can come up with some explanations.
Well, I feel like in one of those, so the mutation rates are higher, but then they actually need more mutations to make the large biological changes that have to happen in organisms, right? Like, one thing I was thinking about is I read somewhere that, like, the prediction of the divergence between the common ancestor between chimps and humans was supposed to be like 3 to 5 million years ago, but it has slowly, like, gone back and back and back to like 6 and maybe now even 7 million years divergence, because they're realizing we have to account for all these large morphological changes. And yet, we don't have time for that with,
it makes perfect sense. I got it. It's hilarious. Yeah. You can only go back so far because of the fossil record. So they only have so much wiggle room. But what you're saying is they're kind of trying to push that earlier and earlier so that they can get more time to affect the changes that they need. I'm afraid that the changes dwarf the time window that they have. A chimp, and it's funny, you look at the chimp and the human and you compare the genomes and you say, well, it's 2% difference. Well, okay, now it's 5 or maybe it's 7. That keeps getting bigger, but it's still, some amount of change that isn't all that great. And then you look at the actual morphologies and the two species and the behavior, it's like wildly different. And how would this possibly evolve from random mutations? It's a great challenge for evolutionary theory to explain.
Well, it feels like a challenge too, because on one hand, they want the mutation rates to be high to get those morphological changes, but then what we see when we get high mutation rates is degradation of the organism. So
it's called mutational load. Yeah, you
okay.
actually you have a limit. That's fundamental evolutionary theory. Yeah, you can't have too much mutation, or else it's going to ruin the whole story. So mutation rates are actually kind of finely tuned. You have to have enough of it to affect evolutionary change, but not too much, because that's just going to degrade, as you said. You'll wreak havoc. You have to have a certain amount of time to absorb these changes, and for natural selection to pick out the good ones, as Darwin says, and to reject the bad ones. And so, yeah.
But if you slow it down too much, then you can't get, then you run into the fossil record problem. So yeah, thank you. Okay, let's see. Optimeg says lots of papers on evolution use abstractions than empirical or concrete verification. This is why chemistry and physics are rarely doubted, but evolutionary biology is often doubted.
Yeah, I mean, you could say that's one reason. I could say another reason that it just, it fails over and over again. The predictions of evolutionary theory just fail. So that's a pretty big reason why it might be doubted. There's also no plausible cause. Evolutionists lack a plausible cause. Every time they try and think one up, it fails. And so we have these debates, that ongoing debates amongst evolutionists, not healthy debates because they don't have a good theory. So there's a proliferation of theories because there's no good one good theory. So if you want to talk about reasons why evolutionary biology is often doubted, that would be a long discussion. There's a lot of reasons, I think.
Yes. Okay. I think, and then Optimeg also says dates from the molecular clock. How wrong can we be? We'll use this paper from now on. Yeah, that's a good paper. I didn't know about that one. I like that. Okay. So Laser Fan, and I think his name is Esteban, if I'm remembering correctly, or Stephen, I don't know. I can't remember. It's something like that. But why would anyone expect there to be a universal molecular clock? That's not anything that'd be expected from biology.
So no, I don't think that's true. And that was expected by evolutionists. The rate of mutation is, yes, if you look at today's evolutionary literature, they'll talk about, it's all over the map. But that's because of the problems that they encountered to fit the theory. In fact, why should the rates of mutation in a gene vary over time, or over the different parts of the evolutionary tree, over different parts of the genome, different parts of the gene itself? This part of the gene, mutated differently than this part of the gene. I mean, that just looks a lot like adding knobs so that I can control the errors here. And it looks a lot like a tautology. Whatever I observe, I'm just going to say it's evolution, and I'll put a knob on that and say, well, evolution did that. I don't see a reason why the mutation rate should be all over the map like that. Yes, you could imagine how, if you had let's say an asteroid come and hit the Earth and create massive environmental damage and maybe degrade the atmosphere. Let's just say I'm making this up. You could cause a difference in the ozone layer, and so the cosmic rays, maybe you wouldn't be blocking out as many cosmic rays or whatever. So there could be massive environmental changes could cause changes in the mutation rate because of the radiation environment. That's just one example. So yeah, you could imagine situations like that. I think that's certainly fair within evolutionary theory. So yeah, does it have to be rock solid? No, but we're at the other end of the spectrum here.
Yeah. Laser Fan says mutation rates between species can vary quite a bit, but this is not a point against evolution. It's always going to be quite difficult.
Yeah. Okay. Difficult. Yeah.
Okay. Going on, dates obtained by molecular clocks will always be hypotheticals unless it's a more recently diverged species like a bacterium in a lab.
Yeah, and so we'd like to tell evolutionists that it's a hypothetical, it's inferred, instead of saying this is observed.
Yes, and the problem that I have with this is how often I'm told we know when these diverged because of the phylogenetic tree and these, the molecular dating and everything. So they're constantly appealing to their genetic divergence dates and demanding that we believe that's when that species, split off. And so, they're treating those dates convenient when it's convenient for them, like absolute dating.
And it's certainly okay, as Thomas Kuhn pointed out, if you're going to have a paradigm and you need terminology within your paradigm, and you can't be questioning your paradigm all the time. You're working within the paradigm. So he called that normal science. And so I certainly understand that and allow for that. And people need to be able to have their own literature and their own terminology and so forth. And so they're going to come off as, yeah, I'm assuming this is true. But when you get into a theory that is so problematic and has failed so many times, doesn't have a plausible cause and so forth, then it becomes, this becomes much more flagrant and much more of a problem. When you're speaking in your paradigm and you're assuming this is true and making these statements that are so obviously problematic empirically, now this is, really, that's why I think we're having this discussion, is because we have a theory that's assumed to be true, which is so obviously problematic on the science.
Thank you. Yes, individual genes will diverge at varying rates. That's why we use multiple genes, so that the general picture is more likely to emerge.
Yeah, and I have a friend who's a used car salesman, and he loses a little bit of money on every sale, but he makes it up on volume. So yeah, we can use multiple genes so that a general picture is more likely to observe. Well, unfortunately, with multiple genes, you get more problems. The more genes you introduce into your multiple sequence alignment, the more problems you have. So it's just this mythology that, yeah, if we get more data, it works out. Inevitably, that doesn't work with evolutionary theory. Remember when they said that when we get more genomes, we'll solve the orphan problem. Instead, it just got worse.
Yes, and I don't think Laserfan was here at the beginning, but Dr. Hunter put up a chart where 4 different genes were compared and the rates don't match. And so, which rate are you going with? I didn't
See, what
see
Laserfan would say there is, yes, the rates don't match, but the trend is the same. So if I compare a horse and a cow on that protein or that protein or that protein or that protein, it's all going to show the same trend as when I compare a donkey and a horse. So you're going to see a similar trend. So they all have those lines that are at least the lines are going up to the right as you go to farther, length of time. Sorry. So,
but wouldn't you
that's what he would say.
always have that? Wouldn't you? Would it be possible to have it go any other way?
No, I think it would be possible theoretically. I think you could possibly have it a different way. The problem is you do have, like, that is such a hand-picked, customized example, and there are so many problems that that chart doesn't show. So that what that chart shows is the first problem, that you have these differences between genes, and the explanation for that was, oh, well, different genes can tolerate mutations differently. Oh, well, that actually fails empirically. So that's one problem. Now we go on to the next problem, So we have all these problems. So that first problem didn't address everything, but it addressed that one problem, that we have these differences between genes and we don't seem to have real good explanations for it. On the histone 4, what they said was, okay, look, what's the purpose of histone 4? What's the function? It creates the hub about which the DNA is wound. what could be more fundamental? What could be more crucial? What could be more sensitive? If you change this, it would destroy all life. Obviously, so they're rationalizing. They're trying to come up with a biologically realistic explanation, rationalization to save the theory. And you say, okay, well, that makes sense. I'm literally reflecting what the literature said. This is what evolutionists said. It's just histones are just so crucial. It had to be that way. Okay, well, that sounds, if I'm an undergrad or high school student, I'll buy that. Except then years later, evolutionists or scientists start testing that. And they find out, no, actually, you can mutate histone 4 and it's not that big of a deal. Uh-oh, that whole thing just fell through now. So the outcomes are not good. You have the failure of the theory and the outcomes are not good.
And he actually chimed in and said 4 genes is still too few. You'd have to use hundreds or even thousands to get a general trend.
Because there's so much noise. You might get a trend, yeah, if you average through. So, if you could apply this sort of reasoning to false theories that we believe are false, like the flat Earth or geocentrism, and you just average through these problems. Mars goes backwards. Well, you can just take enough data and then you average through that. Retrograde motion. Okay, well, whatever. We'll just average through that. So you have to, in science, you have to treat failures seriously. You have to reckon with failures, not just use statistics to, that's the great bane of statistics. It's there's lies, damn lies, and then there's statistics. You can't just use statistics to cover over problems in science that are just obvious. And it's like, we don't have a scientific explanation for this. Your theory is failing. So let's stick with the science, and we have to have mechanisms, we have to have explanations for what we're observing, and we just fail over and over again. That's not a good thing for the theory.
And I am going to go back to earlier comments, but Laserfan is chiming in right now. I'll let the conversation proceed. And he says statistics work that way. The larger your sample size, the more likely you'll find the average.
Yeah, I think that came right out of page 37 of the Stat 101 book, which I have on my shelf back here. So yeah, statistics do work that way. Science doesn't work that way. You can use statistics profitably in science, for sure. It's a very valuable tool, but you have to understand the tool. You don't want to be averaging through or smothering over interesting, important observations. If I dropped this pen and it went up instead of down, one time out of 1,000, I wouldn't want to average that out. I would want to look at that. It's those exceptions. It's those anomalies. It's those weird things. Francis Bacon talked about that, and he was right. You want to home in on those, not average those out.
For example, some human genes may be more diverged than either gorilla or chimp genes, but if we use the whole genome, humans and chimps turn closer to each other.
Yeah, I mean, we're getting into my previous response still applies. Yeah, we just have to be careful with averaging and stick to the science.
Yeah,
I'm not saying don't use statistics. I'm a big stat guy. I've taught statistics. I mean, and I've used it a ton in my work. So I'm a big estimation and stat person. I like using statistics, but I also understand the weaknesses and the how to use them.
Yeah, and I don't know why people use this human chimp gorilla thing a lot. And do you have any comments you want to make about that? Because I don't know.
You're right. That's a real hot topic. Yeah, because I think the primates, human evolution is hot. That really brings out the metaphysics. Because the Roman Catholic Church, for example, says, well, yeah, evolution is fine, but not for the human soul. We have to preserve that. And no, I would say, yeah, you're right. That does need to be preserved, right? Because it doesn't fit the science at all. There's no way. Evolution doesn't explain that for one thing. But there's so many other problems with evolution beyond that. But my point is that when you get to the human evolution story, that suddenly becomes really important. And Philip Johnson talked about that in his first book, Darwin on Trial, and how that really, he has a story about, what was it, the Human Genome Project, or, oh no, it was one of the fossils that was discovered and it was treated so heavily. Anyway, I can't remember the passage exactly, but he has some good material on how when he gets to human evolution, that takes on more importance in the discussion. And for me, I'm more interested in other parts of evolution that are more meaningful to me in terms of how they reflect on the theory.
Right. Yeah. Okay, so let me see. I have okay, Laser Fan says most mutations are neutral. Do you agree with that?
Um, yeah, that's probably true, but I would be careful because, neutral is a hard thing to measure, and a lot of things that appear to be neutral could have an occasional effect. So it's actually, it's just tricky. It's tricky to know these things. But that is certainly kind of, I would say most people would agree that, good mutations are very rare. Bad mutations, harmful mutations are more common, but neutral, probably the majority are closer to neutral, slightly bad, slightly good maybe. But yeah, but just take like a protein coding gene, for example. Most mutations that you apply to a protein coding gene that do cause a coding change aren't going to affect the function too much. So you almost have a neutral effect on function. Let's say it's an enzyme and its enzymatic activity doesn't change that much. But then you do 2 mutations and it's a little bit worse now. And then you get to like 5% of the protein. Maybe it's 7 or 8 mutations if it's whatever the size it is. But it's a few mutations. You get half a dozen mutations. All of a sudden, the performance starts to really degrade. And most proteins can't sustain. You get up to 5-10% of the sequence mutated, and it's not working anymore, or it's significantly degraded in function. And so you say, well, which of those, now, I guess my point is, which mutation was it? Well, it wasn't. It was the cumulative effect. And so you could, if you did any one of those mutations alone, it might have been no change. It was neutral. But together, you had a broken protein. What's my point? It's just complicated. It's just it's more complicated than saying, well, a mutation is neutral and not neutral.
And what you don't have is hundreds of mutations coordinating positively together to bring about novel features in organisms.
Exactly. And this is what evolution requires. Evolution requires large numbers of mutations to happen simultaneously to create a complex new molecular machine or organ of some sort. It's beyond the reach, it's beyond capture for evolutionary theory. It doesn't have the resources to create these things. And so what you have to imagine is that, oh, well, there's some sort of a smooth, gradual pathway of intermediate structures leading to that, but there's no evidence for that. So we don't have empirical evidence for what evolution absolutely requires. So why are we even debating this? That's why there's no plausible cause. The evolutionists don't have a plausible cause, and that's why they're debating so many scathing debates and criticism amongst evolutionists, because they have to try and they're trying to come up with something that works, and they're not able to do that.
Yeah, and not only do you need all those simultaneous mutations, a lot of them, probably most of them, have to be in more than one gene at a time, right? Because most of the morphological features and things are multi-gene, there's many genes that contribute to it. Yeah. And so you have to have simultaneous, not even in just one spot, you have to have it like coordinated across, large stretches.
That is the name of the game. Name of the game in biology. is that you have this sequence of events and different structures working together, whether it's at the morphological organ level or whether it's at the molecular level. Yeah, proteins work in teams. That's why we talk about quaternary structure. Proteins get together and form quaternary structures, molecular machines. And yeah, you often have, if you're going to create some new capability, it would have to have mutations across multiple genes, different parts of the genome, that plus the regulation of it. There have to be other RNA genes that are changed. Yeah, all kinds of things. It's just crazy. It's crazy. It is so insane, the level of complexity and how a random unguided mutations could ever come up with this, no matter how much selection you want to invoke. It is silly. It is a silly idea. You're really, if you are a knowledgeable life scientist, you really don't have an excuse for believing in this. It is so outrageous. It's so silly. And it reminds me a lot of Hans Christian Andersen's story, The Emperor's New Clothes, where you have this guy without any clothes and everyone's saying, yeah, that's a beautiful outfit. We really like that outfit. That's wonderful. And there's this one little boy who says, wait a minute, he doesn't have any clothes on. And it just kind of blows the whole thing. That's what this is. It is so beyond silly.
But, there's one difference in that story, in that the people who were saying that the emperor had clothes on, they were doing it out of fear of being the only one, because basically in the story, you're
Right.
told that if you don't see the clothes, you're not a good person, or, there's something bad about you. Right. And so, everyone's seeing these clothes. But then when it comes to evolution, it really feels like people are so deeply convinced that it's not just something out of, fear of being ostracized or something like that, but they are truly, deeply convinced that it's true. So how do we get to that?
Yeah, I think that's great. It's a great point, Rebekah. I think you're right. The analogy does break down like all analogies. Although I think many people will point out, and I think it is true to a certain degree, there is peer pressure. And within science is a very social activity. And yeah, you need to go along to get along. That is true in science, to get your letters of recommendation, to get your passing grades, to get your entrance to the grad school, to get your Ph.D., to get your faculty position, to get your grants awarded. There is that peer pressure that I think is similar to the story, to Hans Christian Andersen's story. But I also agree that only goes so far. The peer pressure and all that, and wanting to go along with the world, that's only so far, because I agree completely. Most evolutionists, certainly those in print and those who engage in the discourse and so forth, they're not just going along to get along. They are, I agree with your sentiment. They are absolutely convinced that this is true. And
Yeah,
for very, very good reasons.
Well, I'm sorry, go ahead.
And so to your question then is, okay, well, how do we explain that? Is that what your question is? How do we explain it?
Well, actually, I already know your answer to that, which is it's all about God, right?
Yes,
Because
exactly.
it's ideas about God that keeps this afloat. So I already know your answer. But I wanted to point out there. Okay, go ahead.
I need to interject because if someone's dialing in here and they have no idea what's going on and God, what are you talking about? I just have to interject that when we say that it's all about God, knowing that those evolutionists who are making those arguments, are oblivious to the historical theological roots to those arguments and the metaphysical benches that they're leaning on, those metaphysical claims that they're resting on. It's complicated, it's subtle, it's nuanced, it's a deception. And so to them, they actually believe, most of them, I think, actually believe this is just empirical science. We see that in the comments here every week. It's like, evolution is just science. What are you talking about? And so I just want to throw that in, that most evolutionists aren't aware of their own arguments. But you were saying something else.
Well, and yeah, that's important to point out, because that's what I found is actually it totally I don't think people are aware, because they react very strongly when you say that they're making religious argument.
Right.
They cannot see that or fathom that. And
Right.
so I think,
it's a big job. It's a big job to
Not
lay that out. So I say, look, you don't understand that you're resting on religious, and you are actually imbibing and being mandated and being motivated by religion. You don't get that at all. So here's what I'm going to do. I'm going to represent your argument in all of its strength. And I'm going to repeat back to you your argument in all of its glory. And it's absolutely convincing. And I'm going to have you nodding your head in agreement. You're going to be saying, you got it. Yeah, that's it. Then I'm going to dissect the argument. I'm going to lay it out step by step. And I'm going to show the key premise or premises. And I'm going to show the history of that. I'm going to show where it shows up in the 1960s and 50s and 40s and back to Darwin, and then back to the Anglicans and the Lutherans and back to the Greeks, and how it all is there historically and theologically, and how even if you don't know the history, you can at least see you have a premise here. And that premise isn't real clear. It's subtle. It's nuanced, but it's there. And without it, your argument absolutely dies. It is absolutely demolished. You have to have this. And that doesn't come from an experiment. It doesn't come from science. It's an assumption about the world and about God and how they are juxtaposed.
Yeah. And I also wanted to say about the emperor's new clothes, I do think that there is a way that analogy fits perfectly. And that is with people who are not willing to spend the time looking into evolution, but they have just believed it because that's what was taught to them from a very young age. And that's what the smart, educated people believe. So if you want to be smart and educated, let's say, and I've seen this in particular with people who are into philosophy, like the guys who are really into philosophy, usually very smart people, will not spend a minute looking into evolution.
Right.
Okay. They won't even approach it because we already know what science says about that. They don't like the topic, so they don't want to look into it, but they just believe it because that's what the educated people believe, right? But it's very ironic because in like the realm that they're interested in, in philosophy, they'll go deep, they'll investigate it very deeply, but they're not skeptical when it comes to evolution. They will just go along with the crowd.
That's a great observation, because I've seen the exact same thing, but not just with philosophers, but I do definitely, philosophers seem to be common with that. But theologians, I found that with theologians too, that they resting on their biology, their life scientist friends and their life scientist associates. Well, I talked to, Jim, whatever, over at the biology department, and he tells me this. So that's it. And so I think there may be two factors at play. One, a little bit of intimidation. Like, I don't want to, I'm actually not that smart. Like, everybody can understand this, but there's a little bit of intimidation. And the other one is convenience. It's like, yeah, I have those religious beliefs that God wouldn't create this world. So I want that to be the answer. And I'm damn glad that my life scientist friend down the hall is telling me that. And that's my confirmation testing. I'm going to hear that when he says that. And when someone like me says, well, no, that's actually wrong, and here's the reasons, then you're going to do some confirmation testing. You're going to say, oh, well, that's an anomaly, or you're religiously driven. You're an outsider. You're an outlier. You're not normative. Jim over here, he's normative. That's the way it is. You're just trying to nibble at the edges. So I'm going to ignore you. So I think it's confirmation testing for my religious beliefs, or the intimidation thing, or both working together.
Yep. Thanks. Okay. Frank says, excellent presentation on the false prediction of a molecular clock.
Thank you, Frank. Appreciate that.
Yeah, thanks for your support, Frank. Frank also leaves a lot of good comments in the comment section and stuff, so I appreciate that.
Yeah, very appreciated.
R. Brown says, the high scientific standard that you hold evolution theory to, do you apply the same standard to non-empirical sciences like history, archaeology, and paleontology?
So I would if I got into those. Yes, for sure. I do. And I have I do look at the history of thought behind evolution. So yeah, for sure, absolutely, I use that. But I want to chuckle a little bit at the first part of that question, which says high. That word high, the high scientific standard that you hold evolution theory to. I mean, are you serious? High scientific standard? We're talking about a theory that says the world arose by itself. And I'm asking for the scientific evidence for that. And it's failed dozens of times in its predictions, and it has no plausible cause. And it claims to be a fact, an undeniable fact. And if you deny that, you're perverse. And I'm questioning that. And that's a high scientific standard. This is phony science. I don't mean to be negative here. I don't mean to be critical here. But if I didn't use the word phony, it would be just not accurate. With all charity that I can possibly muster here, I have to use the word phony. This is phony science. I'm not a kind of guy that goes around criticizing people easily or criticizing theories easily. I don't think this is a high standard at all. This is a theory that is problematic from the beginning in so many different ways. And that's what we're pointing out, that it makes this claim of being true. How could you make that claim with all these problems? It just doesn't make sense. Oh, well, actually, all the arguments for why it's true are religiously driven, theologically driven. Oh, that's where it's coming from. So it's not so much just a high scientific standard, but it's understanding the evolutionary thought and what it's all about.
Not Evolution says evolution takes too much faith. I agree.
Yep. Yeah, it takes too much faith, or you could say the wrong kind of faith, since those theological assumptions, those theological beliefs that mandate and motivate evolution are non-biblical.
Big Bad Mama says estimates using the molecular clock give dates of approximately 5 to 8 million years ago for human chimpanzee divergence, in general agreement with the paleontological evidence. Coincidence? Well, it's funny. You must have not been here, Big Bad Mama, because this was one of the ones I talked about where the dates have changed. So this was not the first dates that were the molecular dates given for the split between human and chimpanzees. And actually, the molecular date, they would love it to go back further, but they can't because of the fossil record. So this is not in general agreement. You've got to figure out how to get gigantic morphological changes in that period of time, and it's very tough.
But what I like about Big Bad Mama's question is she's making an appeal to empirical study, empirical analysis. Let's look at the theory, look at the data, compare the two, and see if they match up. And here's one that she thinks matches up. We can argue that maybe it doesn't match up as well as she thinks, but okay, at least we're starting off on the right foot. Let's make an empirical study appealing to science, looking at the science. Wonderful. Love it. Now, will we also look at the preponderance of evidence? At first of all, for the molecular clock, that's what we're talking about today, but also all the other parts of evolutionary theory and all the other false predictions we've been looking at, and the fact that there is no plausible cause for evolutionary theory. Will we look at that? So where are you willing to go with that investigation? Are you willing to look at all the preponderance of evidence? If you find there are evidences that fall in line with evolutionary theory, no question about it, but there's not that many. So if you filter the evidence, if you pre-filter the evidence, so you put those positive evidences in your textbook, and you read your textbook, oh look, there's all this evidence for evolution. Those are custom, those are hand-picked. But if you look from a scientific perspective, a theory neutral scientific perspective, you're going to find, oh, there's actually this theory falls apart. So are you willing to go there?
Okay, Big Bad Mama gives us begun DR 2010 Miocene hominids, and
that's the author of the paper. Begun 2010 is the year.
Oh, okay.
And so she's quoting a paper there.
Okay. So, okay, let's see. Okay, this is part of the earlier conversation. So, Laser Fan says morphology can change dramatically fast given the right selective pressures. Do you agree with that?
I didn't know that. That's a new one on me. Thank you very much. Oh, by the way, is there a reference for that? Which experiment did that come from? So no, this is a theory. This is a statement made to support to save the theory. This is not an empirical statement. So morphology can change dramatically fast given the right selective pressures. Okay, so selection pressure is a totally valid, classic evolutionary concept. So that's good. Selection pressure. You're not using it correctly. So that's not how selection pressure works. Selection pressure, it refers to a scenario where you have pre-existing phenotypes, and one of them, and then you have an environmental shift. So pre-existing phenotypes, environmental shift. One of those phenotypes has better fitness, has higher fitness than the other. So it changes the fitness of pre-existing phenotypes. So one now is going to take over or grow in population compared to the other. It's not going to create anything new. Selection pressure does not create anything new. Okay, it just shifts the fraction of the population, or one species does better than another species. So it's a shifting of fitnesses. Morphology cannot change dramatically from selection pressure.
And we do have examples of things changing dramatically, but from what is already present in the organism, right?
Yeah, either from what's already present, yeah. And that could be a structure or design that's already present, or because of epigenetics responding to rapidly. So intelligent directed adaptation to an environmental shift. So within one or a few generations, you have rapid change. It has nothing to do with evolution. That's not evolution. That's not mutations. It's not natural selection. It's directed adaptation, something that evolutionists fought and denied for centuries until they could no longer, because it was too obvious.
And for those who are commenting now, I'm a little bit back far in the chat, and I'm just putting up comments that were put up previously. So I hope to get to your comments in a bit. But this is the kind of follow-up on that one. Why should rates vary? Selection, clearly directional selection and drift as well. So directional selection, is that kind of going against what should be allowed in evolution?
No, it's just selection that keeps on moving in one direction. So this is, again, we're just kind of throwing terminology against the wall and seeing what works here. Drift does not imply that rates should vary. Okay, so drift is a reference to exactly what the molecular clock is talking about. Drift is a reference to random mutations just happening, and you can get, drift and toward, you can luckily go, a protein subject to drift can have degraded or improved function just by luck, just by the chances. So that can happen. That doesn't change the assumption of more or less constant rate.
There are also experiments with bacteria in which bacteria evolved a faster mutation rate in real time.
Well, when I assume we're referring to bacteria that have been subjected to a different environmental shift. So yeah, you have in the experiments where you subject bacteria populations to a shift in the environment, a challenging environment, Some bacteria will they will mutate at a faster rate, and then there will be some bacteria, some individuals in that population that actually go hyper. They go at a much faster rate. So that's a directed change. That's a change in the way that the individual is operating in response to the environmental shift.
So the organism itself already had that capability. It's putting to use a function that it has to be able to deal with different environments, right?
Right.
It's like, so it's what it already has. It doesn't have to do with evolution.
Right. It's not going to save the molecular clock problems.
Yeah, Mike says, I'm a bit late, but please tune in. Thanks, Mike. Appreciate your support.
Yeah, thank you, Mike.
And Optimeg says, lots of problems in molecular evolution models. A new paper came out about the bacterial flagella, which showed that the stators are motors that power the main motors of the flagella.
Yeah, I saw that paper. I have not had a chance to read it. That looks like a very interesting paper. So I would like to take a look at that. But I mean, the whole flagella is a great example of just mind-blowing complexity that people can kind of relate to it because it kind of looks like a machine. So it's a good example. And evolutionists attempted to explain how it evolved by pre-adaptation. So you had the different parts were there for other purposes. And then they luckily came together and formed this flagella. And it's a very, just so story, just a serendipity, invoking a lot of serendipity, which evolution often relies on. Oh, it just happened that way. This incredible thing. They don't really understand statistical mechanics, the way the universe actually works. So it's just drawing upon these super unlikely scenarios to solve the problem.
Yeah, and the more that I find out, it's like everything that you find out about the way that life functions, it's just another mind-blowing experience of, wow, this is incredible. It takes so much for, there's so many processes that are dependent upon each other and little molecular machines that are dependent upon each other. And so if you didn't need it at one point, then what was the evolutionary benefit of getting it later, right? And if you need it for your essential functions, then, and what you end up with is a web of, thousands of things that are dependent on each other. And if even one fails, the whole process can fail. Topoisomerase fails, the whole thing fails. You can't go on without all of these things working together. Maybe you could lose a few and still function, but there are some things that are absolutely essential and they're dependent on each other.
These are great points. Great points. And I love the initial point you made. If it wasn't needed before, then why is it needed now? I mean, those problems just come up over and over and over again. And when it is needed, there's all these other players that has to play with. Where did they come from? Were they there or are they not there? If they weren't there, then this isn't going to help any. If they were there, then they weren't going to work without this thing. It's just these problems occur over and over and over again in evolutionary theory.
And just at so many different levels, even just in one little cell, even the simplest cell, it's just, evolution has no hope. It needs to be abandoned. And that's what it amazes me how much confidence people have over the theory, that they're so sure that it's true. And many of them have studied biology very deeply. And that's what's like, I understand the way that your understanding of it, but it doesn't fully explain it for me. It's just, I'm still like, but how?
Right. It is shocking, really shocking when you sit through a lecture and somebody with a PhD in grad work, and he's been a professor for 40 years, and he's going through, homeostasis or the ribosome or the mitochondria or the electron transport chain. And it's just kind of mind blowing. And he's just, and it evolved this way and it evolved that way. And so I think there is a deception at play here at some level.
Yeah. So Laser Fan says as long as the structure of the protein or the main sites of the enzyme are conserved, you can have a huge amount of substitutions and sequences to enzymes which are functionally the same.
Yeah, that's tricky. That's real tricky because it may be true that if you are careful and you select the right residues, you can mute those to your heart's content, and you can collect all of those. So for a globin protein of 140 residues, there may be dozens and dozens that you can play with, and you're not going to have any effect. But you just have to know which ones. And if you're randomly choosing, you're not always going to get those right ones. But it's tricky because it's hard to, the space is so huge. We don't know how it performs over this entire space. If you look at the different globin sequence, I'll go with that example because that's well studied. You can find different globin sequences that are quite different, like down into the twilight zone. So like 20% or 15% similarity between two globins. That's like, almost completely different. But they both are globins. They both have the same function. They look the same. The fold is the same. They have the same function in different species. But is there an evolutionary pathway from one to the other? Can you mutate this one to get to the other one? And if so, how particular is that pathway? Those questions we really don't understand fully. And so it's not really clear, what you're claiming here is not that clear exactly how that works. But we do know that random mutations applied to a protein does not end well very soon. But yeah, if you apply selection, you might be able to get it to only do the mutations that don't do any bad thing. Are you going to go to some other place? Are you going to form some other protein? Can you evolve another protein that way? That's a tall order.
Optimeg also put in about a dozen motors powering the main flagellar motor. For this to emerge is impossible.
Yep.
And she also says over 50 proteins needed for the assembly and function of the bacterial flagella. The cell is a small volume with thick viscosity too. So how this would evolve is unlikely.
Yeah, I mean, absolutely. It's really, we're as skeptics of evolution, we're in this funny world of trying to show what is the probability of something happening that's super low probability. These astronomically low probability events. So it's not an easy thing to show. Like, how ridiculous is this? That's not that easy to quantify. It's not like the simple problems of an urn with balls in it or something.
Tyler says, what definition of religion is Hunter using to include science within it?
So I'm not including science within it, first of all. So evolution is not science. Evolution is a religious theory. The definition of religion, that's a fair question. What do you mean when you say it's religious? What are you talking about? So I'm not, and it's true that people will use nuanced definitions like, oh, that's religious, So I'm not using a nuanced definition. My definition of religion or theology is claims about the attributes of God. If you're making claims about God, claims about God's attributes, last time I checked, that's religious, right? So I think we can all agree on that. So this is not heroic. I'm not doing anything nuanced here. I'm not being picky. Oh, that's metaphysical. You talked about uniformity. That's metaphysical. No, I'm not doing that. We're nowhere close to anything nuanced here. It's nuanced in the sense that they don't use the word God. You do have to understand history and philosophy to see that. So yes, it can be deceptive, but the premises, the theological premises in evolutionary theory are very significantly religious and theological. I don't think there's any debate about that. When you make claims about God that can't be discovered through experiment, there's no experiment you can perform to learn these things. Well, that's religious. We can agree on that, can't we?
So base skeptic, I guess, says, examining origins back to spreading lies. Just stop this nonsense. And following on, it amazes me that you are still trying to pretend there is any real debate about the fact of evolution. So I realized that for you, evolution is a fact. But like Dr. Hunter likes to ask, why is evolution a fact? And what amazes me is that so many people are so amazed, but they can't bring any evidence for evolution. So bring the evidence for evolution. Let's stop with all the arguments about whether what I'm saying is true, or about like evolution is true, blah blah blah. Just show why it's true. Like let's just drop all the back and forth and accusations and just show why it's true.
Yeah, I mean, that definitely is a great point, Rebekah. And we can also bring up the incredible world that we live in. Look out the window, look at the flagella we were just talking about. You just look at these things, and then you look at the claim that this is a fact. This is evolution of this is a fact. The world must have, no question about it, evolved by chance, by random, unguided forces. The world arose by itself. It arose spontaneously. Those are different ways of saying the same thing. And then you have comments like this. So clearly we're in a situation where we have a theory that produces false predictions by the dozens, does not have a plausible cause. They can't come up with a explanation. So they're fighting amongst themselves, coming up with multiplicity of explanations. And then you have the claim that it's got to be true. And so do you see the disconnect? It's got to be true, but yet it fails over and over and over again. So what am I missing? And then, of course, what you're missing is it's true because of the theology. And so when you have people like, base skeptic here saying, it amazes me you're still trying to pretend that there's any real debate here, that falls right in with this mode of argument of delegitimizing the science, delegitimizing the empirical evidence, and holding the theology that you're resting on has to hold sway. And so that's just this is the argument you get from religious people. This is the argument you get from theologians.
Yeah, and I realized I was reading the name wrong. It's be a skeptic. So be a skeptic. Please be a skeptic about evolution. How much time have you spent actually looking into this and figuring out if evolution is actually true? Have you challenged it?
So, Rebekah, you came up with the question, what's the evidence? Give me the evidence. What you're making the high claim that it's true. So what's the evidence? That's a great approach. However, I did that. And so I started in on this many, many, many years ago, and I collected every argument and every evidence I could find for evolution to reconstruct the case. I wanted to make the case for the fact, for the truth of evolution, or even that it's just likely, whatever, every positive thing I could get. So I answered that question, and I came up with a list, and it's a long list of all these different arguments. And some of them are, of course, more obvious, more common, used more commonly, and are more powerful. And they're not science. They're not from science.
Wow, I want to go through your list sometime. I think that would be a great show.
Well, yeah. Okay, yeah. But that's basically my books and my papers and stuff, yeah.
Yes, and Dr. Hunter does have two great books. Darwin's God. Wait, is it Darwin's God?
Yep,
yes,
yep,
Darwin's God. And
Science's
Blind Spot. Thank you. And I love both of these books. They're really awesome, and they're very eye-opening. So highly recommend those. Now, so incredulity isn't, this is a comment from QK. Incredulity isn't proof of evolution. Proof evolution is false, Rebekah, but it does take incredulity to believe it's true.
So this individual sounds like kind of a middle of the roader. Some doesn't sound like an evolutionist, but
I'm confused by this comment. I don't
yeah,
understand this
I'm
comment.
not 100% sure either.
Okay, yeah. Okay, so you're saying, okay, okay. Yeah, I'm not sure. Explain it to us. Okay.
I think what is worth commenting on here, however, Rebecca, I'm sure you've run into this argument many times. I have, where they cast your skepticism as incredulity.
Yes. Oh, every day. Every day.
Very common.
Yes.
Okay. So you just can't believe that there's one little problem with the theory, so you just can't believe it.
Yes. And now BA Skeptic says evolution is the foundation of modern biology. These people are liars. Now, I understand why someone can have that perspective, because if you go to a biology class, on every few pages of your biology book, they're going to say evolution connection, and they're going to start talking about evolution, and the entire thing feels like it's all interpreted through the lens of evolution. So I can see how somebody might feel that way, right? When you get done with that. But what you need to do is realize that that's a lens that you've been taught to see through, and actually pay attention to what the actual evidence for evolution being true is. And you'll find that it's severely lacking.
Also to look at, okay, you just said look at the evidence for why evolution is actually true, but also how it plays a role in biology, because the comment here is that it's foundation of modern biology. So you could ask the question, okay, how is that true? What role does it play? Because I think you're right, Rebecca, if you take a class, it's easy to come out of that class, or if you're in a lab and you're working under a professor and it's all evolution, 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 it's easy to get that mentality and to get that kind of belief that, hey, this is just foundational. I mean, we talk about this all the time. And every time we write a paper and every time we come up with a new result, we understand it in terms of evolution. Like evolution is foundational, but it actually isn't. It's always a gloss. Evolution is a gloss that you're applying. Like, how is evolution foundational to the citric acid cycle? Oh, it isn't. Well, how is it foundational to the mitochondria? Oh, it isn't. How is it foundational to how the brain works? Oh, it isn't. You put a gloss on it, though. So Phil Skell was a member of the National Academy of Sciences, a professor, a chemistry professor at UPenn, I believe it was, and he sent out a very short survey to his associates and life scientists around the country. And he had two questions in this survey. One question was, do you believe evolution is, true? Something like that. And the second question was, do you use evolution in your theory? Does it impact your science? Do you use it in your research work? How important is it? Something, I can't remember the wording exactly. And the vast majority of responses were, yes, I believe it's true. No, it doesn't impact my science. So this claim that it's foundational to modern biology doesn't hold true, not only from Phil Skell's survey, but also if you really dig in and understand the science and understand the our modern life science knowledge. What is the state of the life sciences today? How do we understand biology and all of its richness and everything? And what role does evolution play in that? It's always a gloss, and it's always a difficult gloss. It's always like you having to like squeeze the theory and change the theory. So it's not true. That first sentence there is not true. Now the second sentence tells us a lot, though, right? These people are liars. So it always comes to, for rationalists, it always gets personal. You always get the value judgments. They bring the value judgments to the table. There's got to be something evil about you. So lies need enemies, right? Lies need enemies. In order to propagate a lie, you need a bad guy to shift the attention to, right? We're the good guys, they're the bad guys, us, them, tribalism, and focus on them. So if you're in a court of law and the lawyer is trying to convict a guy and it's kind of a weak case, how does he argue it with the jury? He focuses on how evil the crime was and how terrible the crime was. It was, this person got killed and everything, and this was terrible, brutal, and not talking about the evidence of why this guy did it, but rather just focusing on the terribleness of the crime. What does that do to your sense of self-righteousness, your sense of justice? Like, yeah, let's get this guy. You're guilty. Wait a minute. What was the evidence that he did it? Oh, yeah, that's really weak. So you see the importance of having an enemy and having a bad thing to focus on, the enemy to focus on. And that's what this is. This is a very important part of evolutionary theory, is the other, the bad guy, the evil one, the other theory that's false. And that's, of course, what the whole theological basis is of evolution, is creationism or natural theology or intelligent design or whatever you want to call it. It's that other idea that there is teleology. It must not be teleology. So we're focused on that. And so whenever you talk about evolution and the problems of evolution, they don't want to talk about that. Get the light off of evolution. Get it on the other guy.
Yeah, I just wanted to bring this up. I had to pull this book off my shelf. So this is sort of like it's called Biochemical Evolution. It's sort of like a textbooky, book on evolution. And it's really interesting because he devotes so much time to speaking against creationism. And I find a lot of that in these evolution books. I mean, in Francis Collins' book, he spent a considerable amount of time on this, like, creationism, intelligent design. And it's just interesting because it's like, if this is so scientific, if evolution is so scientific, why do you have to worry about that? Right. Like,
right. And so what you need to realize is from the very beginning, it starts from the very beginning, the very origin, the genesis of Epicureanism and evolutionary thinking. It comes out of a rejection, a rebuke, a negation of creation, what we call creationism. And so if that's false, then this has to be true. Okay, so let's work on this now. So let's get evolution going. So evolution didn't come about and then by the way, creationism is false. No, that was, that's the genesis. That's the motivation. God would not have done this. God would never have done this. God would never have designed this world. He wouldn't have created this world. He wouldn't involve himself this way. So there's all these different religious theological arguments about why God wouldn't have done this. Therefore, evolution must be true. So evolution comes second. It comes after the rejection of creationism. So it's embedded, it's inherent, it's at the very beginning, these religious arguments and claims. So people like be a skeptic is a religious person, is a religious, deeply theological person.
And so be a skeptic. We do just want to, again, challenge you. What would happen if evolution as a theory disappeared from the world today? Like, what would happen to science? What would be different, right? Like, would medicine be different? Would, our understanding of how, systems work in biology, like, would any of that change? Like, what would be different? Like, what would we lose if evolution was gone as a theory? And I mean, I really don't see how we would lose anything if that disappeared as the framework for how we understand life. Do you agree with that, Dr. Hunter?
Oh, absolutely. Not only do I agree with it, but evolution has held back science. It has harmed science over and over and over again. So it has caused problems because of interpreting evolution, interpreting biology in terms of evolution has held things back. And we had some examples of it actually in this podcast.
So
so for being a skeptic, what would it take for you to why would you call someone a liar who says maybe this wasn't evolved, I think evolution is a problem. This doesn't work very well. Maybe there's teleology. Why is that person a liar? So think about that. Okay, go on to the next question.
And also from BA Skeptic, irreducible complexity has been thoroughly debunked.
Yeah, so there's no citation for that. There's no reference. There's no such thing. This is a talking point. Of course it hasn't been thoroughly debunked. That's a joke.
I've never heard it debunked. I've heard people say it's debunked, but
I've seen papers. Yeah. I've seen papers against it. The evolutionists have written papers attempting to debunk it, and other evolutionists cite that as, oh, see, they debunked it. So there are journal papers about that.
Okay, just not effective in debunking it.
Right.
So tell us, BA Skeptic, tell us about the conspiracy. Why are the scientists lying? We don't think they're lying.
Well, not only are they not lying, they're actually making valid arguments. So the arguments for evolution are perfectly valid. I guess if you want to talk about a lie, I mean, there are times where it gets aggravating and you just think, come on, how can you say this is, this or that? But I always come back to, this is what they believe. And so I wouldn't call it a lie typically. I mean, there are times where it gets aggravating, but yeah, they're working, they're religious, they're theologically driven people. It's like kind of saying, is a creationist a liar? I mean, I wouldn't call them liars. They are operating from a particular theological conviction that they have, and they're interpreting the evidence from that perspective. I guess the difference in evolutionists is they do not typically acknowledge their theological convictions. They use them and they show up in their text and in their presentations, but then they turn around and say, this is just science. So I think of that as an internal contradiction, that they're confused and they're deceived. I don't normally think of it as lying. We've talked about that earlier in this podcast, but it does get aggravating, yes, that you would wish that they would be like creationists and acknowledge their theological convictions.
And just to be clear, we're not saying there's any conspiracy involved here at all. There's no conspiracy.
Right.
It's just how we got here is a long process, but it's not a conspiracy.
Right.
Okay. Tyler says evolution isn't divorced from science just because Dr. Hunter says so. We need a valid argument for that. Tyler, we've made several shows on this, but beyond that, Dr. Hunter has written actually 3 books on this topic. One of them I haven't read yet. But so, he has spent a lot of time laying papers. Yes, journal papers as well. Go ahead.
He said, we need a valid argument. Okay. Is that what you need? Are you really telling the truth there? I'm not doubting it, but I just want to make sure you're serious about your comment. Because if you're serious about that, we have the valid arguments. We have a lot of valid arguments. You can watch my videos. You can watch Rebekah's. You can read my books, my journal papers. There's a lot of valid arguments. So they're there. If that's is that what you needed, was a valid argument? Because I've got papers. I've got playlists on my channel. I have a paper on why evolution is a theological research program. You could read that paper. It's got valid arguments. So if that's what you need, then they're there.
And I just want to say, Tyler, I'd be happy to buy you both of Dr. Hunter's books that I've read. And so if you want those, send me an email. I'll buy them for you. I'll buy them for several other people too. If I get emails up to 5 people who want to read those books, I will buy those books for you. So anybody who really does want to hear these valid arguments, happy to provide those.
And just to say right up, to take the question even further, evolution isn't divorced from science. So what do you say about dozens of false predictions that don't matter? When you have a false prediction and they do not have good outcomes, they have bad outcomes. increased complexity and sweeping it under the rug and so forth. So if it wasn't divorced from science, why wouldn't those matter? How could you still, and you don't have a plausible cause for evolution, you have dozens of false predictions. How can it then be a fact if this isn't divorced from science? And then what about all the religious claims? How can it, if it isn't divorced from science, why are there all these religious claims? How can evolutionists make all these theological claims and then say, well, no, we're not divorced from science? So I guess, to me, the valid arguments are really, really obvious.
BA Skeptic says, lie, lie, lie. I love this because this is just showing, look, BA Skeptic, what have we lied about? What is the lie? What have you identified as a lie?
Yeah, it's unfortunate.
Thank you all there's a lot more comments here, but I think we're over 2 hours now, so we need to shut it down. But, I really enjoyed this conversation. Thank you for being willing to come on and share your thoughts. I really like interacting with people in the chat. And so it makes the conversation fun. So we appreciate you. We appreciate the people who support, and we appreciate the people who come on and challenge, because we are here as people who want truth, and we want to help people see the truth. And we don't want, anything hidden. We want, what is reality to be out in the open. And that's all I'll say as a final thought. Did you have something you wanted to share?
I think those are great points, Rebekah. I thank you for inviting me, and thanks to everyone for coming on. As Rebekah said, challengers and supporters alike. For those challengers, it can be very deceptive, and I keep on coming back to Hans Christian Andersen. It is such a telling story that he wrote about the Emperor's New Clothes, because we are talking about the world arising by itself spontaneously as a fact without any scientific support for that. When I say not any scientific support, I mean, just there's so many problems. I should say that, put it that way. There's just so many devastating scientific problems. So what would it take to think about, to accept that? I think it's very difficult for people to accept that. So thanks again to everybody, and thank you, Rebekah.
Yeah, thank you, Dr. Hunter. And so for, this, we went over one of many false predictions in evolution, and we intend to continue just going through and showing you the false predictions made by evolution and the problems with evolution. So thanks for joining us, and God bless you.